if you have questions or concerns, feel free to stop by. So it's every Wednesday between 1:30 and 2:30. So essentially after this lecture. And so between my lecture sessions, so until April 1st. So my office is in 5471. So again, feel free to stop by, or if you have questions, if you want to arrange a different time to meet, feel free to shoot me an email. So personally, I work in the field of bacteriology, microbiology. So I do a lot of microscopy and also a lot of mathematical modeling to study the dynamics in bacterial cells. So it's pretty exciting to be here to introduce you the next couple topics for this course. So regarding the topics that I'm going to cover, so for today and part of next lecture on next Monday, so we're going to talk about the human microbiome, which is a very hot topic nowadays. And then we'll talk about afterwards. We'll talk about how to control the spreading of microorganisms in the environment. So what chemical and physical tools we have to contain the growth or proliferation of microbes. And then we'll talk about the antimicrobial chemotherapy. So basically, the biochemical mechanisms by which how antibiotics work. And then towards the end, so we'll briefly touch on the microbial genomics. So it's essentially some sequencing techniques for us to identify and quantify the composition of mostly bacterial cells in the natural environments. So you notice like the schedule is somewhat different from the ones that's posted on Canvas. Because I decided to include during the very last lecture, I decided to include some new topics or new findings in the field, because as someone who works in the field, I just couldn't resist the temptation to share with you some of the exciting discoveries that are made during the last several years, because they're having some really, I would say, significant milestones in the field of microbiology that were made just during the past several years. And it's more, and none of these, these topics will be on your exam, so it's more like a enrichment or for eye-opening experience. So similar to the first part of the course, so we don't require textbooks, so these chapter numbers are just for reference. I can guarantee you whatever that's going to be covered on your final exam will solely be based on the materials that are shown on the slide. So especially those terms that are highlighted in green, as you will see. So these are the core concepts that I try to highlight and make it clear. It's very, it's something that you're supposed to know. Okay. Yeah. So any general questions or logistics before we get into biology? Okay. No. Okay. So before we talk about microbiome, I just kind of want to motivate sort of this notion that microbes are everywhere. It occupies every single conceivable ecological niche on the planet, and always been there. It's been there pretty much since the beginning of the Earth. So you can see from the slides, the Earth formed about 4.6 billion years ago, and the earliest life form, which is bacteria, appears, pretty much soon after, right? 3.5 billion or 5 billion years ago, and since then it's been the most abundant, the most adaptive. Yes. Oh yeah, good question. So slides will be uploaded after lecture, or actually, do you prefer me to upload it before the lecture? Before lecture? Okay, I'll try to do it next time. Okay, cool. Okay, good. Any other questions? Okay, I'll upload the lecture slides before the lecture next time. Okay. Yeah. So since then, the bacterial cells have been probably the most most successful form of life, and in terms of adaptability and in terms of like diversity, that's ever been identified. So it shouldn't be surprising that for it's the microbes have been living with us for as long as you can imagine. So we humans are colonized, inside and out by bacterial cells, and we have been essentially we've been co-evolving with bacterial cells through the entire evolutionary history of our human biology. So we rely on bacteria to perform a lot of different functions because there are a lot of I think it's of no exaggeration to say that without bacteria, we humans cannot even sustain our own life because there are a lot of essential physiological functions for which are absolutely critical for sustaining life, but we just don't have a genome in our own. We don't have a genome to perform these physiological functions. For example, if you want to break down complex sugars, right? We don't have, a lot of times we don't have the gene that's necessary that's encoded in our own genome. So we rely on these bacterial cells or microbes to perform these essential functions. So we humans can be thought as a sort of like a super organism, right? So it's like a mixture of microbes and mammalian cells. And for this reason, we humans are known as holobionts. It's just a word that means it's like a conglomerate of microbial and mammalian cells, which exist and also co-evolve over time, and to form a single functional units. Just to clarify some terminology here, because sometimes they're kind of used in a sloppy way in daily or colloquial language. So microbiome, so there are two definitions for microbiome. So first one is broader definition is refers to the entire collection of microorganisms in the host, along with the genome and elements from the surrounding environment. So that means the broader definition means the refers to the cells, microbial cells, as well as the genome and RNA, protein metabolites, primary, secondary metabolites. So everything that's associated with the microbes, so living in a higher organism. So that's the broader definition of microbiome. And then there's like narrow definition, which just refers to the collective genomes of microbes living in a host. So essentially that's a DNA, microbial DNA. So typically, it's pretty self-evident in terms of which definition applies whenever you see these words from literature or from news. And then there's a closely related but different word, which is microbiota. As a lot of times, in colloquial language or even in scientific reporting, sometimes they're used interchangeably with microbiome, but in practice, it shouldn't be. Because microbiota has this very specific definition, so it refers to the community of microorganisms living in a specific environment. So whenever you're talking about microbiota, you sort of have to specify like what kind of ecological niche. For example, when you're referring to the microbes living in your mouth, we use the word oral microbiota. And once in a while, you're gonna see this word microbial flora, or microflora. This term, I don't know why it's still being used all the time in news media. It's like totally obsolete term. So we're not gonna use this term in this lecture. Essentially, this term basically it was invented during a time when people's thoughts, bacteria are the same. Belongs to the plant kingdom, which is not true, right? So we're not going to use this term. So just know the definition of these two terms. And probably from the first half of the course, you sort of have an idea of what constitutes the human microbiota, right? So the bacteria obviously is one of them, and then there's archaea. I think there are a few species living in our guts. Fungi, which is mostly it's yeast cells. Protists, for example, we have some algae cells that are living on our skin. So virus, So in terms of like absolute abundance, virus is like the most abundant uh, species. But again, for this course, we're not gonna talk too much about the virus because there's like separate field study which specifically studies the composition of the viral species in human microbiome, which is called a virome. So for most of the course, we're going to be talking about bacterial cells, which is like the next most abundant and probably the best studied components of of the microbiome. Whereas the virus is largely understudied nowadays. Uh, Maybe let me give you a quick mini-mirror question to see how much you know about the human microbiome. So by the way, this is how we take attendance, at least for my portion of the course. Uh, so please pull out your phone and scan the QR code. So try to make an educated guess or try to make some guess to see if you have a good sort of impression about what's the abundance of bacterial cells living in our like own human body. So how does it compare to the population of Hong Kong, which is about 7 million, right? 7 to 8 million. Uh, or is it comparable to population on Earth, which is about 8 billion, over 8 billion? And number of stars in the galaxy. So that's on the order of 100 billion. So 1 billion is 10 raised to 9th power, right? Number of hydrogens or a number of hydrogen ions or protons in a bacterium or a number of these. Okay, so let's see what your guesses are. Okay, so most of you have chosen number of stars in the galaxy. By the way, do you have any idea in terms of how many hydrogen ions or how many protons are there in a single bacterium? Is it going to be larger than this number or is it going to be smaller? This is a kind of digression, but I think it's some number that might be interesting to know. So the number of hydrogen ions. Any guesses? Is it going to be larger than the number of stars in the galaxy, or is it going to be a smaller number? Okay. Is it just based on some calculation, or okay? Any different guesses? So actually, if you are curious, you can do this. It's a very simple backup envelope calculation, right? If I tell you the pH of the cell is 7, right? So you can convert pH of 7 to concentration. So now you have concentration, and if I tell you the volume of a single bacterium is about 1 micrometers cubed. So you have concentration, have a volume. You can just calculate the absolute number of protons. So it turned out to be a really really small number. It's less than 100. So sorry, this is digression, but I think it's some number, biological number that's just totally goes against our intuition. Um, so so the correct answer is actually none of these. Yeah, about uh, so here's the number. So number of stars in galaxy, as I mentioned, it's about 100 billion. But number of bacterial cells in a typical human body is on the order of trillion. So trillion is 10 to the 12th. It's a very staggering number, right? So I hope this gives you a quick impression in terms of how abundant these species are. Um, Okay, maybe try the next question. Okay, now you know there's a lot of them in our body. But okay, so how does it compare to the number of mammalian cells in our human body? Right, you typically think of us as humans as you know mostly comprised of mammalian cells, right? So how does the what's the ratio between between bacterial cells and mammalian cells in our human body? By the way, can I ask if this ratio has been taught during the first half of the lecture of the course? No. Okay, so most of you think it's 1 to 10. So for every mammalian cell, there's like 10 times more bacterial cells, right? Um, so that is a number that's widely circulated in the literature, in the news media, and this is a number that people believe to be true for the past several decades. But it turned out this number is not correct. So so it turned out so you probably heard from the news media or reading from some online sources or literatures saying it's a ratio bacterial cells outnumbers our mammalian cells in our body by a ratio of 10 to 1. So this ratio was actually derived from a very rough calculation that was done in the 1970s from a very old study. And several years ago, I think almost 10 years ago, this number has been revised from a new study. So the correct answer should be should be one roughly equal. Uh, yeah, so this number has been revised due to some new research progress that's been made during the past 8 or 10 years. So we in our human body we roughly have the same number of bacterial cells compared to our mammalian cells, um, but it doesn't really minimize the importance of bacterial cells in terms of like the physiological function, as you will see during today's lecture and next week's lecture. So here are the numbers that I think you should be familiar with. So so this is like a number of human cells and mammalian cells. We have about, don't worry too much about these specific numbers, 30 trillion, right? Bacterial cells, it's about 40 trillion cells. So you can see it's comparable, right? It's slightly outnumbers our mammalian cells. So the ratio is about, you can just know it's roughly one to one ratio in terms of like mammalian cell versus bacterial abundance. In terms of like the body weight, it doesn't really contribute that much in terms of like body weights. It only contribute to like 2 kilograms roughly to our body weights, and the vast majority of these bacterial cells are actually found in our gastrointestinal tract or guts. And it's extremely diverse. So far, there's been over 1,000 bacterial species that are being identified and sequenced in our human guts. So if you consider like the typical, a typical bacterial species has about 2,000 to 3,000 genes. So if you multiply that 2,000 to 1,000, so that's gonna give you 2 million bacterial genes in the guts, microbial genes in the guts, compared to us human, we have about 20,000 human genes. So in terms of like the diversity of genes, or gene encodes biological functions, right? In terms of like the diversity of potential biological functions that can be performed in our human body, it's the bacterial cells vastly outnumbers the number of genes or potential physiological functions that can be accomplished using our own mammalian cell genes. So this kind of provokes a question of it's like a philosophical question, right? Are we humans really humans, or are we some kind of like a hybrid system? Uh, in terms of like not just in terms of the composition of cell types, but also in terms of like genes, in terms of potential physiological functions. So, uh, and these are some of the numbers I found online. So I think most numbers are up to date. So 100, yeah. So there are over 100 trillion microbial species, including bacterial species. And as I said, 90 to 95% of the microbial bacterial species or microbial species actually are located in our guts, GI tract. And yeah, so as mentioned in the previous slides, the number of bacterial microbial genes vastly outnumbers the number of mammalian genes in our human body. So the reason why our guts can host so many microbial species is because it has this really huge surface area. I think this number is like it's like overestimate. So it says the surface area of GI tract is the same as the size as 2 tennis courts. This is like overestimation. So our surface area of the guts is about half the size of a badminton court, but still it's pretty substantial, right? So that's why it's huge. It can host like many microbial species. Yeah, it's about 1.3 times more than the number of mammalian cells in terms of like sheer number. So that 10-to-1 ratio is outdated. 2 kilograms, as I mentioned, in terms of like contribution, in terms of body weights. Yeah, so probably the only time I'm going to mention about the viruses. So the viruses outnumbers bacterial species by a factor of roughly 5 to 1. So most of these viruses are bacteriophages. So these are viruses that eat the bacteria, which should make sense, right? Yeah, this is some interesting facts. So if you purify all extract all the bacteria from your guts, and if you just analyze them up, head to toe, the sheer length can actually wrap around the entire Earth, the equator, for more than twice. So it's a very mind-boggling number. Yeah, and each person has a unique gut microbiota, pretty much like how fingerprints works. So these are some interesting figure to know. So in terms of what these bacterial species are doing in our body, well, so here is a scanning electron micrograph of a for a mouse, for a gut sample from mouse. So stained in green and blue are the intestinal tissues. So the cell nuclei, essentially the mammalian cells, part of the mouse gut intestinal GI tract. And stained in red is the bacterial species called Bacteroides fragilis, and in yellow is E. coli. So you can see there's a lot of them, right? And also they coexist with each other, and also they live in close physical proximity to the mammalian cells. So there you can see they live together. So we use the word symbiosis to describe the relationship between these microbes. So symbiosis in Greek just means to live together. So in general, there are 3 types of symbiotic relations. And so the first one is called commensalism. So commensalism just means you have species that just lives in with the host and without causing any harm to the host, but it benefits from its association with the host by finding a suitable habitats and also access to nutrients. So, for example, the Staphylococcus epidermidis, as you can guess probably from the name, epidermidis means on the skin, right? So it's a species that's found on the skin. and mucous membranes as well. So it just sits there. It doesn't do much. It doesn't do much to the host. But we secrete nutrients, and they're gonna feed on them. So that's called commensalism. And then next relationship is called symbiotic relationship. It's called a mutualism. So basically mutualism means both the host and the microbial species will benefit. For example, E. coli, it'll help us to break down carbohydrates and other nutrients in the guts, so they can be utilized by other microbes and also the hosts. They also produce vitamin K for the host, so which is important for help with blood clogging. I think E. coli also produce vitamin B, so which is important for brain and heart functions. You probably read from news, last year there was like a physician in Hong Kong, doctor in Hong Kong who got killed by infection of E. coli due to food poisoning. So you probably have this impression that E. coli is pathogenic, but actually most of the E. coli species living in our guts are non-pathogenic. There are a few pathogenic species, but they're like the minorities. So most E. coli's they engage in this they have this like mutualistic presence in sort of a symbiotic relationship with the hosts. And then the last symbiotic relation is parasitism. It's pretty easy to understand. So for example, a good example would be the influenza virus, so H1N1. So we'll talk about what H1N1 means towards the end of my session. So it's like it's like virus, right? So it infects the host and hijacks the molecular machinery to to replicate, to produce more copies for themselves. In this specific example, H1N1 would infect the respiratory epithelial cells without providing any benefits to the hosts. But it's actually can be detrimental, right? Because the severity of the infection is largely dependent on the how robust the immune function of the host is. It can be anywhere between having pneumonia to even death. So these are three symbiotic relationships that I expect you to know. Okay, what it means, and specifically what it means. But also in terms of like the duration of the occupancy, the symbiotic relation, the microbes can be also classified based on the duration of their occupancy on the host. So they can be broken down into resident microbiota. So these are species that permanently colonize the hosts, and they're mostly commensal. So for example, the Staphylococcus epidermidis mentioned in the previous slide, it's just something that sits on our skin. It's not going anywhere. It's always there. And then, but in contrast, there's like something called a transient microbiota. So it'll occupy like the host for anywhere between like few hours to several months, and eventually they're gonna be wiped out for a number of reasons. So for example, due to competition, right, for nutrients with the other microorganisms that are also present in hosts, and also can be cleaned up by our immune system, and also the chemical and physical change. Microbes are very sensitive to chemical and physical changes, so they can right, if skin, for example, on our skin raises the moisture level and the acidity, and could change, which could favor or disfavor the growth and proliferation of certain species. So these are the two general categories of microbiota based on the duration of their occupancy. And so this is a study that was done, I think, 2 years ago by our neighbor Macau. So it's basically it took like over 1,000 fecal stool samples from different continents, and actually it was done over a decade ago. So and they revealed this staggering number of almost 10 million microbial genes. So as I alluded first, non-microbial genes is just vastly outnumbers the human genes. And another important finding from the study is that they found most of these microbial genes, they're not silenced, so which means they're actively transcribed. Um, which is important, right? Because if it doesn't mean anything if you just have a gene that stretch of DNA or if it's not transcribed. So that means that they're actively perform a lot of different functions that may not be easily uh, that may not be easily that may not be feasible with our own mammalian cells genes. So for example, for digesting food, metabolize drugs, and priming our immune systems, for example. So in terms of like, so you can see, okay, I hope we can appreciate the diversity of the microbial genes. But the question is, okay, where do we get our microbiome? When for a long time, people thought, when a baby is born, like it's born sterile. But it turned out that not to be true. So people have found that newborns are actually they're born with microbes from the immediate environment. So, for example, if from the mother's birth canal or vaginal canal, if the baby is born naturally or by the skin, of the initial caretakers, nurses, doctors, and so on. If born by a cesarean delivery. So here's the question. So if infants are born, by cesarean delivery, they don't really get to inherit the microbiome from the mother's vaginal tract, right? So still, 60% of the infants' total microbiome comes from the mother, despite they're born by cesarean delivery or C-section. Any guesses? Where do these microbiome come from? How do this infant obtain the microbiome from the mother, despite it's born by C-section? Any guesses? Yes. Yeah, that's right. That's one major mechanism. So by physical connections. But there's actually another major route that's probably more important. Any guesses? Other guesses? Yes. Yeah, that's right. Yeah. So that's a major mechanism. So if a baby is born by C-section, so the vast majority of the microbiome can be delivered to the baby through breastfeeding, breast milk. Um, so actually, over 700 species are identified from the breast milk. The single most important species that you have to know is this one, Bifidobacterium longan infantis. I guess from the name, right? Infantis means infants. It's something that's predominantly found in infants. It's not only the most abundant species in infants gut microbiota. It's also so it typically accounts for about 50% of the infants' GI tract, the gut microbiota in infants. The number is slightly lower in developed countries because of the use of antibiotics and so on. Anyway, so it's not only the most abundant one, but also functionally, it's definitely one of the most abundant one, most important one because it actually does a lot of. It's important for an array of different physiological functions for the development of the infants. So here I'm just going to list a couple. So the first one is that the bifido infantis can actually convert fructose to acetates. So what it does, essentially, the acid when it gets converted to acetates, the acetates will coat the surface of the epithelial cells. And what it does is that once the epithelial cells gets coated by acetates, the acetates will help to tighten up the junction the junctions between the epithelial cells. So basically, it'll reduce the permeability of the epithelial cells. So the epithelial cells can be better at fending off the infection of foreign agents. Um, and also it can help to prime the immune system to prevent epithelial inflammation and cell death in the lower column caused by toxin. So, for example, so for one type of pathogenic E. coli shown here, it can produce a toxin called Shinga toxin. But if the column is epithelial cells on the column is coated with this bifidobacterium infantis, coated by the acetates that's converted by the B infantis, the Shinga toxin cannot penetrate this coating, so it'll try to avoid triggering severe immune responses. Maybe just another word about this name. It's kind of mouthful, bifidobacterium, but bifido means so it comes from the prefix of bifurcates. So bifurcates means form branches. So as you can see from the schematic diagram, so it has like this Y shape, right? It looks like a letter Y, so it means bifurcates from branches. So that's where the name comes from, bifidobacterium. Yeah, so this is true for B. infantis, whereas it's not true for a closely related species called Bifidobacterium adolescentis. So this bacteria, the closely related strain, cannot convert fructose to acetates, so it cannot coat the epithelial cells. Okay. And besides this function, so this bifidobacterium infantis is also very important for the neurological development of babies. This is a fairly new finding. I think it's just a couple years old. So people have found that the B. infantis actually can release sialic acid, which is a metabolic byproduct. Essentially, this sialic acid will try to tune the immune system of the baby, and so that the immune system can actually prune or trim extra synapses that are unnecessary. So essentially, this study was first done in mice. So people have found that for those mice which did not have which were raised germ free and did not have this species presence in their guts, these mice form way more neural connections, synapses that were necessary, which is bad. B. infantis, actually helps to prime the immune system to trim down the extra synapses. And also, it's also required components of brain glycolipids, so it's like essential components, the sialic acid. Yeah, and so what exactly is there in the breast milk that that's important for the development of this baby? So the most important, the most important components, one of the most important components of breast milk is called human milk oligosaccharides, so HMO. So it's like third most amount of components of human milk, so after lipids and lactose. So essentially, human milk oligosaccharides or HMO contains over 200 non-digestible and non-nutritional carbohydrates. So when people first were analyzing the composition of this human milk, and they found the large presence of the HMO, people were puzzled because how come you have so much of this HMO in the human milk, but none of the stuff is digestible can be utilized by humans. So people were kind of puzzled back then, because it appears to be of no nutritional value to the human. Any guesses in terms of why this might be important? Yeah, that's right. Yeah. So it turned out the bee's infantis really likes this. So the bee bifidobacteria infantis actually feed on this HMO. So it's kind of so HMO is kind of a prebiotic, what's referred as prebiotic. So prebiotic just means some substance that promotes the growth or beneficial microbes. Okay. So we humans cannot digest it, but the beneficial microbes can utilize it and produce something good for the hosts. Yeah, so HMOs creates environment which allows bee infants to flourish and outcompete the other species that initially colonize the infant's gut but cannot metabolize HMOs. So the idea is that even when a baby is newborn baby just inherits like a tiny fraction of bee infants, which is vastly outnumbered by the other competing species. But if through breastfeeding, you're preferentially promotes the growth of bee infantis. So it's simply just due to the competition between species. After a while, the bee infantis will begin to be the dominant species, to become the dominant species, because you have their favorite food present in the breast milk. Okay, maybe I'll take a 5 minute break for you to refresh your mind before I continue. So let's come back at 12:50. Okay, before I continue. Okay, let's continue. So as I said, so when the baby is born, it's predominantly dominated by this bifidobacterium infantis. But over time, the composition will change of the human microbiome. Um, so the idea is that so a lot of factors will actually play a role in deciding what the composition of the microbiome would be like for each individual. So such as the birth process, it's natural birth or by C-section, and nutrients use of antibiotics and so on. So the general trend is that the diversity of composition of microbiome will typically stabilize after H3. I mean, H3 and onwards, I think it's still somewhat plastic. Um, but once you reach like age 18 or 20 something, it's really beginning to solidify. Um, and so the composition of the microbiome once you're an adult, it doesn't change that much until you reach age 60, 70. That's when you begin to see a decline in the diversity of the composition of microbiome. Um, and of course, like if you just look at like shorter time scale on a day-to-day basis, I mean things can change. Sometimes can change dramatically. For example, if you're using antibiotics, right, that's gonna kill like a large proportion of the bacteria in your guts, or if you're having diarrhea. So on a shorter time scale, you still can have some substantial change in your own microbiome composition, but overall, the composition is pretty robust in the sense that even if it gets disrupted, it can recover typically. So after a while, for example, a week after you're having diarrhea, you can pretty much regain the signature microbiome composition that's typically present in your guts. Um, any ideas in terms of how this can be accomplished? So, right, if you use antibiotics, you're going to kill many of these bacterial species, but after a while, you can regain them back. Yes. Yes, yeah, that's great. That's one major mechanism. So there's something called the core, what's known as the core microbiome. So these are the ones that are highly resistant to antibiotic killing. For example, they can form spores. They can enter dormant states, so they're not vulnerable to the attacks or cells of antibiotics. Uh, and you probably heard about something called a horizontal gene transfer. So for these survived species, they can pass the gene which encodes the antibiotic resistance to neighboring cells, even for different species. So over time, you can have these bacterial cells, gaining back the ability to proliferate despite the presence of antibiotics. Yes, that's one major mechanism, and of course, there's physical through physical contacts. You can absorb the antibiotic the microbial species that are typically present in your gut, by food or physical contacts, social contacts, and so on. Yeah, so after age 3, especially after adulthood, it's pretty much solidified on a longer time scale until you reach become a senior person. But in contrast, the as I was mentioning about the gene expression pattern, right? The gene expression pattern is more strongly influenced by the local changing local environment. So the combination remains largely the same, but if whether certain genes are being actively transcribed, it's contingent on it's more, I would say it's more plastic. um, in a sense that can be easily influenced by the local environments. Uh, yeah, for example, diet, infections, states, sex hormones, and external environments can influence which genes are expressed, which microbial genes are expressed. Yeah, so many different factors can either promote or destabilize the microbiome. So genetics, that's something we have no control of. Essentially, genetics will play a huge role in shaping the local chemical environments of your guts, for example, in terms of, I guess, typical acidity, right? Uh, Viscosity and so on. So that's something we have no control of. First of all, we already mentioned about it. Siblings, so there's like this interesting, so I would say it's kind of becoming more and more debatable because people, I don't well, so there's like this hypothesis that if you have more siblings, you're gonna have healthier microbiome because you just simply because of social interactions you can gain more diversity in terms of like kind of microbiome microbial species you have access. It's kind of I don't know, it's kind of debatable. Seasons, infections, antibiotics is pretty self-evident. They will influence the state of your microbiome health. Smoking, so smoking is really bad for the health of your microbiome. You probably saw it's bad for your lung, right? But it's also that's terrible for your microbiome because smoking will acidify your guts. So a lot of these pathogenic bacterial species they actually prefer a slightly acidic environment. Uh, yeah, so they change the pH of your guts. Um, yeah. So what exactly are these species present in the body? Um, so these are four major phyla that I expect you to know. We can go over them one by one. So the vast majority of the gut microbiota belong to these two phyla, so Bacteroidetes and Firmicutes. So these two together account for over 90% of the bacterial species, bacterial phyla in the guts. Uh, we can briefly mention about, I mean, the names are a little bit mouthful. So Bacteroidetes. So so what does bacter mean? Maybe this will help you to better memorize the names. Bacter means bacter means rod. Looks like a rod shape, right? So uh, oides just means like, resemble or like. So it looks like a rod. For example, shown here is like a picture of the Bacteroides, which is like a species from this Bacteroides. So you can see it's like a rod shape. So it's gram-negative and includes species such as Bacteroides, Promotida. You don't have to worry about which species belong to which phyla, but you have to know the phyla name. Yeah, and also together with Bacteroides, which comprises a majority of the human gut microbiome, is Firmicutes. So again, firm means strong, right? Robust. Uh, Cutis. What does cutis mean? Anyone speaks Latin? Uh, yes. Yeah, exactly. You speak Latin, right? So strong skin. So as you can probably guess from the name, it's gram-positive, right? Strong skin refers to this phylum has a very thick cell wall. So thick cell wall means it's gram-positive, right? It's more likely to be gram-positive. Yeah. So for me, these gram-positive species, including these we'll touch on some of these species later. Lactobacillus clostridium, which is a really nasty pathogen. Staphylococcus, streptococcus, and so on. And then besides these two, there are the other two major phyla that I expect you to know. So proteobacteria, which includes our old friend E. coli and Helicobacter pylori. So proteo, what does proteo mean? Any guesses? It's from Greek. It's actually from a Greek god called Proteus. Right. So it's a god which Greek god which can assume many different forms, shapes. So for example, shown here is a picture of the Helicobacter pylori. So you can see it's like a spiral shape, whereas E. coli is raw shaped. So this basically this phylum, the name indicates that phylum can has many different shapes. Can also adapt to many different environments. So it's like a very high diversity sort of like a phylum. You probably see a lot of like this. Some of you, this might be for familiar species because you know go if you go go a lot of times you go through routine health check a hospital, you're gonna do this medical test to see if you're affected by the Helicobacter pylori. It's one of the few bacterial species which has been categorized as classified as a carcinogen, which means it can cause cancer. So it inhabits in, in our guts, and it can trigger inflammation and cause gastritis. And if you have prolonged gastritis, that's going to predispose you to stomach cancer. So that's why it's one of those carcinogens. Um, yeah, and the last one, actinobacteria. Again, actino in Greek has a meaning which means speaks Greek. So it means radiating or star-like. So as you can see from the image, it's like it looks like a star, right? Radiating or so it's a very characteristic of so this kind of shape is a very characteristic of bacterial species from this phylum. It's quite positive, so it includes Streptomyces. So this is like the most dominant species within this phylum. Corynebacterium, Propionibacterium, and so on. So again, make sure you know the names of these for dominant species, dominant phyla. Most of these bacterial species in our guts are anaerobic, which means they either don't require oxygen or they just cannot survive in the presence of oxygen. So the ratio between anaerobic and aerobic species about 1,000 to 1, which makes sense, right? Because there's in our guts, especially in the lower part of our guts, there's not much oxygen, or there's no oxygen. So that's another reason why it's kind of challenging to do experiment with these, to study human microbiome, because if you're wanting to work with species in a lab, you have to work in anaerobic environments. So you have to set up, a specialized anaerobic chamber, which will eliminate the presence of oxygen. Yeah. And yeah, any questions regarding these four species for phyla? So I'm going to mention a little bit about more about these first two. As I said, physiological function-wise, they perform many different physiological functions. So I'm just going to introduce a few. So to basically to as sort of like introduction in terms of what they do in our human body. So so turns out bacteroidetes and firmicutes are involved in weight gain or obesity. So this finding was first discovered in experiment involving using mice. So first people found so mice which were genetically predisposed to obesity had more firmicutes and fewer bacteroidetes than compared to mouse that were lean and healthy. Yeah, so this is something that you should know. So high fat, high carbohydrates favors the growth of firmicutes, whereas low fat, low carbohydrates favors the cultivation and proliferation of bacteroidetes. So the way I try to remember this is to use the first layer. Firmicutes corresponds to fats, right? And bacteroidetes, B is slimmer or bonier. Um, yeah, so people first found there's this association between the ratio between these two phyla and the obesity status in mice. Of course, this is pure correlation, right? It doesn't tell you, it doesn't establish a causal relation. So people went on to perform experiments to try to see if it's indeed the presence of these two species that are causing the mice to become obese. So they did a couple experiments. The first one is so they compare the weight status of mice of germ-free mice, which means the mice that were born and bred in isolators, so they were born and raised sterile, so they don't have any microbiome. To conventional mice, which means a mice which has microbiome. So they gave the germ-free mice and conventional mice the same diets, a very high-fat diet, and they try to see which one, if which one will gain more weight. Any guesses? Are the germ-free mice going to gain more weight, or the conventional mice tend to gain more weight? Why? That's right. Yeah. So yeah, you're right. Uh, yeah. Conventional mice gain more weight, whereas germ-free mice gain less weight. I mentioned it earlier. So a lot of these bacterial cells they help to break down those nutrients. So this, of course, this doesn't really tell us, okay, is which species is responsible for the weight gain, but at least it establishes this causal relation between the presence or absence of microbiome and your tendency to gain weight or become obese. So that's the first experiment they did, um, and then they did another follow-up experiments. So so they did this microbiome transplant experiments. So where the gut bacteria from the obese mice were transplanted into germ-free mice without changing the diets. Uh, Essentially, after they transplant the gut bacteria from obese mice to the lean mice become fatter. Um, Again, this is another further corroborates the notion that the microbiota is indeed playing a role in weight gain. And then, uh, yeah, so germ-free mice transplanted with gut mice from obese mice put on more weight. Yeah, and so basically, this experiment tell you that okay, the germ, those obese mice, something in the microbiota of those obese mice is triggering this weight gain, right? And then they want to find out which species are. Okay, so sorry. So here's like how this fecal transplant is done in mouse. So they just force this mouse to eat the poop. Not eat the poop. It's like injecting through the nasal passage. So if you want to see how this is done, there's like movie tutorial. So you can check the link. I guess most of you haven't had lunch yet, so I don't want to show something disturbing. But if you're curious, you can check out the movie here. So it's gonna teach you how to do this experiment step by step. But essentially, the mouse is forced to sniff the poop the other mouse. And then so importantly, so mice and humans have different microbiota, but the two largest divisions are shared. So Bacteroidetes and Firmicutes are both the most predominant phyla in both mice and humans. And people have found that even if you just transplant the you know the stool sample from human to the mouse, it can replicate what has been observed with fecal transplant between mice. So, for example, if you transplant a stool sample from a obese person, the mouse will tend to get more. Whereas if you transplant the poop from a lean person, if you transplant the bacterial cells purified from the fecal sample from a lean person, the mouse stay healthy and lean. So yeah, so people through a bunch of studies, people have found that if, even for people, so this is no longer just true for mice. So later people found out even between two people, the one who hosts more firmicutes than the Bacteroides tend, even if given the same diet, so it's like controlled experiments, right? The one that has more firmicutes would be heavier. Of course, as I said, our genes, our genetics plays a role in terms of what kind of uh, bacteria we tend to, our gut environment tend to favor for proliferation. For example, I myself personally, I'm very prone to gain weight, so I suspect that somehow my genetics predispose me to host more Bacteroides, to host more Firmicutes. So does that mean we're hopeless in losing weight? No, because you can take probiotics. So that's one major mechanism by which you can sort of manipulate the status composition of your gut microbiota and change it and tip the balance. And if you don't want to, take prebiotics or probiotics, you can also just change your diet. So this is like a longitudinal study that was done, I think, quite a while ago. So people just follow like the weight gain and also the composition of gut microbiota for people who have been switched to healthier diets. So after 52 weeks, so that's a year, right? Essentially, these people who were obese, they after they switched to a healthier diets, they began to the composition, the ratio between Firmicutes and Bacteroides become look more similar than the ratio more comparable to the ratio as found in lean people. Yeah, so lifestyle matters a lot. And in contrast, if don't don't overuse antibiotics because it may trigger obesity by wiping out good microbes. Of course, along with bad ones, and consequently disrupting the natural composition of gut microbiota. So let me just emphasize. So it's a ratio. It's not an absolute number of these two species that matters in dictating your propensity to gain weight. It's a ratio that matters. So besides these two phyla, I just want to mention another one it's very recent discovery. So this species called Christensenina. So people have found that people who eat this Granny Smith apple, they tend to host more of this species Christensenina, which and thin people, leaner people have more Christensenina in their guts than obese people. I think. Yeah, it's been verified through fecal transplantation. So people have found that transplanting this bacteria to mice made them slimmer. So another thing I want to mention regarding this is that sorry, let me digress a little bit. So the entire field of microbiome research is very new. I don't know if you're aware of this. So this research really began to take off. I think it's over the last decade or so. And there's like over the past decade, there's like a huge explosion in terms of like these findings such as these. So my personal advice is that whenever you're seeing claims such as eating this microbes will have this beneficial results, try to take it with a grain of salt because this entire field is in its infant stage, and there's a lot of financial or economic incentives which I wouldn't be surprised to to motivate some to cause exaggeration in terms of claims they're trying to make. I think this particular study has been well established, at least like causal effects, but just be mindful whenever you see some sort of advertisement because there's just investors are pouring so much money into this field nowadays. So regarding these fecal transplantation. So it was first those kind of transplanting bacteria was first done not in the context of like a weight control. It was done to deal with the more severe scenarios. Sorry. Let me just first go over why do microbiota affect the body weight. The way that these microbial species can influence on the body weight has I mean, there's so many different involves so many different physiological processes. But here I'm just getting three major mechanisms. So as I said, gut microbes helps to break down dietary fibers, complex sugars, the monomers, and thousands of enzymes in the guts involving such process have been identified. And so these microbes not only break down the sugar into monomers, it also help to ferment into something called a short chain fatty acid or SCFAs, including the three major types: butyrates, propionates, and acetates. So here's how it works. Butyrates, shown here. Basically, it's preferentially absorbed by the epithelial cells, and it's like a major energy source for the mitochondria in the epithelial cells. You know, mitochondria is like energy house, powerhouse, right? So essentially, it feeds to the mitochondria, these epithelial cells in the column. So the epithelial cells in the column are called colonocytes as well. So it helps to fuel the colonocytes mitochondria to convert the carbon substrates. So that means the food that you eat into carbon dioxide. So essentially, butyrate helps to basically vaporize the carbon source. So that's how it helps to lose weight, right? Because once it converts to carbon dioxide, it will just be gone. The second one is propionates, which is like a salt form of the propionic acid, which you probably learning about chemistry. So basically, this travels to the liver. It gets delivered to the liver, and it inhibits the cholesterol synthesis. So you can see why it's beneficial. And also, I didn't mention here. Butyrate and propionates together also help to suppress the secretion of hormone, which to help promote the secretion of hormone, which will suppress the feeling of hunger. So it'll suppress it'll make you less likely to feel hungry. So that's why these two are good for weight control. Acetate is a little bit different. So its acetate will get converted to fatty acid and triglycerides in the liver as well. And so that's actually gonna be because it gets converted to fatty acid and triglycerides, which means your body will store these fatty acids. So which means acetate, as opposed to the first two, it'll make you more prone to gain weight. So it's a little bit different, opposite results. Yeah, and the preliminary, the causal relations between these have all been verified using fecal transplantation. And maybe I'll just briefly talk about how fecal transplantation was used in dealing with antibiotics resistance next time. So I guess I'll stop here, and I'll see you on Monday. Okay. Oh, maybe one last. Let me just give you one last Mentimeter question for this. It's very simple. Very simple question to test your short-term memory, I guess. It's asking about the without human microbiome. All right. Okay. Bifidobacterium is predominant species in infants, right? Not in adults. So to make sure you know